So now I'll go through the steps that Carolina talked about and what can go wrong and how we can detect it. So the first and very critical step, as Carolina said, is to dissociate cells. If you're lucky, you're working with blood cells or something that's already dissociated, and then it's not a big issue, but many of you want to go into different tissues, and tweaking these dissociation steps to work with your specific tissue is also very important. It's not one method that works for all. And of course, one problem might be that you have incomplete dissociation, you have cells sticking together. If you dissociate too harsh, you can damage the cells. And this is also often quite biased. If you have a heterogeneous tissue and you dissociate it too harsh, it might be some cell types that are more sensitive, that break, and other cell types are happy and viable. So the cell type composition you get out when you do your single cell RNA-seq might be biased due to your dissociation protocol. And then, of course, if you get a lot of broken cells when you dissociate, or a specific cell type that breaks, a lot of RNA will leak out of those cells and be in your suspension. And you might get that background into all your droplets or wells or whatever method you're using. It's also been shown, it's actually a thing that hasn't been studied so much. I know of this one study, they basically did dissociated and non-dissociated tissue and compared gene expression between them and showed that there are a lot of genes that gets upregulated during these dissociation conditions. So it's actually a fact that you might introduce some bias in your data by doing dissociation that you should be aware of. And the only way to really know this is to do a bulk RNA-seq in parallel, right? So if you have genes that are high in your single cell RNA-seq, but not in your bulk, they might also be artifacts from dissociation. It might also be a lot of, I mean, you introduce apoptosis, a lot of apoptosis signals and so on that come up during dissociation. And also, as I said, you might get a lot of this ambient RNA, basically RNA leaking out into your suspension. So this is some data that Paulo analyzed before. It's immune cells done in 3 different batches with different time points. And what you can see clearly in this green time point, you have a lot of neutrophils here, but you also have genes that are this marker for neutrophil expressed in pretty much all cells from that batch. Oh, sorry. So this is quite likely one of these ambient RNAs that leaked out and end up everywhere. And these might be batch specific, or they might be in all your batches. It's not easy to know. There are not that many tools to deal with this, but there is this SoupX package and also some other ones where you basically take these empty droplets that you discarded before, those that only have a few detected genes, and you check what are the genes I detect in these empty droplets. That will give you a feeling for what sort of background RNA you have. And sometimes you see very clear signals. Like here is hemoglobin, you probably have red blood cells. But it's tricky because, as I said before, depending on how this knee plot looks like, what you call empty drops might actually just be red blood cells and not empty wells. So then maybe you need to put your cutoff even lower and rerun the program. In this example, you see a lot of B cell genes in the background. So that's most likely that you have a lot of broken B cells for some reason in your sample. Then the next step of the procedure is to capture single cells. And as Carolina talked about, there's multiple methods. You can do the manual steps, you can do fact sorting, droplets, microfluidics. Pretty much all of these methods will give doublets and most likely some empty wells or just cell debris in a well and so on. And you might get biased selection for certain cell types. I think Carolina already mentioned that with these droplet-based methods, there's a bias against very large cells that you might lose. And it's also important to keep the time for getting to your single cells short so you don't damage the cells. Then the next step in the library prep, once you have them in your droplets or in your wells, is to lyse them. And actually now most people just run with these kits, but as we had some questions about plant cells and so on, then you might need different lysis conditions to keep your RNA intact. What you also should keep in mind that depending on the cell type and the lysis conditions you have, you may not have nuclear lysis. So that will clearly bias the mRNA content you get out. Like some nuclear link RNAs will be quite abundant. The intronic read fraction will be quite abundant if you also capture all the RNA in the nucleus. But in some other cells, you might not lyse the nucleus, and therefore there will be a bias between the populations. And then the next step is reverse transcription. I think we covered this quite a lot. I mean, this is where we get most of our dropout rates. And also it seems it's really unclear why, but it's been shown in a lot of studies that each library prep seems to have different dropout rates for some reason. That's really unclear. Then of course we have the amplification step, and of course that introduces bias, but the UMIs will in most cases control for it. So we can hope that it handled most of this issue. Of course, the chance of detecting a transcript that has a high amplification bias is higher. So at the low end of detection, also these UMI-based methods will be influenced. And then finally, we do sequencing. In most cases, you would assume that sequencing doesn't introduce any biases, but there might actually be issues with sequences that influences, like if you have some contamination in one sample that affect the flow cell and so on. There might be issues with index swapping between cells in the indexing in the sequencer and so on. So it's not perfect, but it's probably one of the smaller contribution to biases in our setup.